The World Federation for Hemophilia has produced a set of guidelines for the management of haemophilia. This is the 3rd edition. The guidelines are available to download for free on the WFH website and also in the journal at the bottom of the page. My name's Megan Sutherland. I'm a clinical scientist and I work at the Genomics Diagnostic Laboratory at the Manchester Centre for Genomic Medicine in the UK. I'm going to describe to you the chapter 4 of the guidelines for haemophilia, and that's the genetic assessment chapter. I have no competing interests to declare. The authors contributing to chapter 4 for the genetic assessment are Carlos de Brazy, Barbara Conkle, Srimati Shetty, Glenn Pierce, and Alak Srivastava. Genetic assessment of haemophilia is important to define the disease biology and identify the genetic variant that's underlying the disease. It's important in establishing the diagnosis in difficult cases, for example, where the disease phenotype in an index case is not known. And it's important to enable the identification of a genetic variant to predict the risk of inhibitor development, and also can identify variants that might respond to different therapies. Genetic assessment is important to identify carrier females and at-risk individuals within a family that's carrying haemophilia. And by doing that, we can provide prenatal diagnosis. The WFH has a number of recommendations about who should be referred for genetic testing for haemophilia. For those individuals who have suspected or established haemophilia, then we would recommend testing all of those individuals for either factor 8 or factor 9 genetic variants. Prior to screening, we'd need to do phenotypic screening in order to determine the factor 8 or factor 9 coagulation levels, so we can determine which gene is involved and which severity of haemophilia we are looking for. After we have identified the underlying disease-causing variant in the index case, we can offer testing to obligate carriers and at-risk females within those families, which can then be used as a tool for prenatal diagnosis in the future. For symptomatic females who have low phenotypic coagulation factor 8 or factor 9 levels, then we can offer genetic testing to identify if there is an underlying genetic cause in the factor 8 or factor 9 gene that is causative of these levels. In these individuals, as well as doing the factor 8 and factor 9 coagulation phenotypic levels, we would also want to screen for the von Willebrand factor antigen and von Willebrand factor activity levels, as this could aid in interpretation of downstream genetic results. Prior to offering genetic testing to any of these individuals, we'd need to undertake detailed genetic counselling, and this should be done by a genetic counsellor if available, or if not, then we could be a clinician who has a knowledge of haemophilia genetics. The purpose of the genetic counselling is to obtain informed consent from the patient, their parent or legal guardian, and to provide information about the genetic testing and provide education about why we are doing it. The genetic counselling should describe the experimental limits of the molecular results and should also include that we might not find a genetic variant. The possibility of incidental findings in other genes, depending on the methodology used, should also be described during the genetic counselling session. The approach to genetic testing for haemophilia varies between different individuals that are referred for genetic testing. In the index cases, when they are referred for genotyping, we want to know their underlying phenotype. This can divide them into different groups, which will then inform the downstream processing for genetic testing. In patients who have severe haemophilia A with a factor 8 coagulation level of less than 1 international unit per deciliter, or those patients with a low borderline moderate level of 1 international unit per deciliter, we're going to process these patients in a particular way. For severe haemophilia A, up to 45% of cases are caused by the factor VIII intron 22 inversion variant, and this is a common underlying variant in these patients. A further 2% of these severe cases are caused by the factor VIII intron 1 inversion. So when we have a case of severe haemophilia A, we're going to screen for these inversion mechanisms first. We will also screen those patients with a low moderate level of factor VIII of 1 international unit per deciliter. If the intron 22 or intron 1 inversion screening is positive, then we will repeat this on an independent sample using an independent time of that method. And then we will report those results back. No further analysis will be taken on these samples. So it's sufficient to just screen for the intron 22 and the intron 1 inversions, as this will be the cause of the haemophilia in these patients. If these inversion mechanisms aren't detected, then we will cascade those samples on for further analysis of the factor VIII gene. We'll do full factor VIII gene screening. So factor VIII is located on the X chromosome. It consists of 26 exons, so we will screen all 26 exons, the splice site boundaries of the exon and the intron, and we'll screen for the 5' and 3' untranslated regions. By doing this, we can test for small structural variants in the factor VIII gene that are causing the haemophilia A phenotype. We'll be looking for small variants, so single nucleotide substitutions. We'll be looking for small deletions, small insertions or duplications, and we can look for frame shift mutations as well in this way. If we find a small variant, then we will be able to confirm that by independent analytical process. That can either be done by the same methodology or an independent methodology. Once that's confirmed, then we'll be able to report those results back to the clinician. If we don't find a small variant in these patients, then we will go on to look for copy number variants. And we can do this by a variety of gene dosage techniques. And I'll come on to talking about techniques in a moment. If we find a copy number variant in these patients, then again, we'll be able to go on and confirm those and then report those results back. The copy number variants are looking for large deletions or duplications in the factor VIII gene. If we don't find a copy number variant, so we've gone through for the severe patients through the intron 22 and intron 1 inversions are negative. We're negative for small variants in factor VIII gene. We're negative for large variants, large copy number variants in the factor VIII gene. Then we haven't been able to identify a variant using the current methodology. And we could predict that there would be a deep intronic variant within factor VIII that might be causing their disease phenotype. And using some methodologies, we are able to detect deep intronic variants, for example, massive parallel sequencing or whole genome sequencing. But these techniques aren't available in all situations. So this is for the group for the severe haemophilia A patients. For those with mild or moderate haemophilia A, so the moderates are 1 to 5 international units per deciliter, and mild haemophilia A is greater than 5 international units per deciliter, then we would go for full factor VIII gene screening in the first instance. These group of patients don't have any common mutational mechanisms associated with them. So we would look for the full factor VIII gene of the 26 exons, the splice site boundaries, and the 5' and 3' untranslated regions. And again, we would follow the flow chart looking for the small variants. Then we'd look for copy number variants in these individuals, so looking for large deletions or duplications. For the mild, moderate, or severe haemophilia B patients, severe being less than 1 international unit per deciliter of factor IX, moderate being 1 to 5 international units per deciliter of the factor IX coagulation factor, and mild being greater than 5, then we would look for the full factor IX gene screening. Again, there are no common mutational mechanisms associated with factor IX, so we would look for the small variants first of all. The factor IX gene, again, is on the X chromosome. It consists of 8 exons. So we would do sequencing for the 8 exons, the intron exon splice site boundaries, and the 5' and 3' untranslated regions. Again, we would look for the small variants, so the single nucleotide substitutions. We would look for small deletions, insertions, or duplications. If these were positive, then we would report back that we found a factor IX gene mutation that was associated with the factor IX phenotype. If we didn't, then we'd go on to look for copy number variants, so small or large deletions or duplications in the hemophilia B gene. The copy number variants can range from a single exon to the full gene. So those are the groups that we've just described are for the index cases. And once we've identified the variant in an index case, we can then go on to screen obligate carriers of those individuals and also at-risk females in those families. If we know the underlying variant in a family, then we will just look for that variant when we do family studies. So we're looking for the familial variant. So we're just going to do a targeted screen in these individuals. If this targeted screen is positive, then we will be able to say that they are a carrier of the familial variant for hemophilia. And this is either hemophilia A or hemophilia B. In these individuals, if we don't find the underlying familial variant, then we'll take different strategies. So for example, if this was a mother of an index case where we hadn't found the familial variant, then we couldn't, then we need to consider mosaicism in this individual. She might be a germline or somatic mosaic for the particular mutation. So she might not carry the mutation in her peripheral blood cells, but she has that in her germline cells. But there is a slide later on considering these areas where we don't find the familial mutation or we don't find a mutation in a particular individual. The methodologies that we can use to screen for the different variants in factor 8 or factor 9 can vary widely, and there are many different methodologies that are available. In the screening for the intron 22 and intron 1 inversions, we could use, for example, southern blotting, long PCR, or inverse shifting PCR. And when we are choosing a methodology to use, we need to be aware of the sensitivity and specificity of the approach to detect the target. For screening for small variants, we can do PCR and Sanger sequencing or next generation sequencing or whole genome sequencing, for example, to detect these small variants. But again, the resources that are available to us may indicate which testing that we do. And the approach and use of a specific technique will depend on the available technical resources. And in places where these resources are limited, it may be appropriate to do a cost-effective screening approach prior to Sanger sequencing of the full factor VIII gene. For detecting copy number variants, we can use techniques such as MLPA, which stands for multiplex ligation dependent probe amplification. It's a gene dosage technique. And we could also use next generation sequencing to detect these large copy number variants. But again, it depends on the available resources within your area. Other considerations for techniques when choosing the analytical techniques, as well as the sensitivity and specificity of the approach that is used, we need to be aware of the turnaround time for producing an interpretive report within the expected time for that test. All results need to be confirmed by independent analytical testing of the DNA sample, and that is either by repeat testing or by an independent method. One of the indications for genetic assessment is to enable the facilitation of prenatal diagnosis for families where it is indicated and where it is required. It's important to be aware of and follow the relevant laws governing genetic counselling and pre-implantation genetic diagnosis and prenatal diagnosis in the country where the services are being provided. The reasons for prenatal diagnosis are for to terminate an affected male fetus if that's desired. We can use it to guide the management of the delivery of the baby and to predict the risk of inhibitor development and the response to immune tolerance induction in the affected individuals. So to do prenatal diagnosis, we need to know the underlying variant associated with factor VIII or factor IX in the pregnant female carrier for the disorder. In females that we don't know the underlying variant, we wouldn't offer prenatal diagnosis. Counselling of these individuals should include a discussion of the risk of the PND procedure to the pregnancy. For example, it could induce early miscarriage, or it could induce early labour. Prenatal diagnosis can be taken either in early pregnancy by chorionic villus sampling or by amniocentesis, or it can be done in late pregnancy by late gestation amniocentesis. During the process of taking the sample for prenatal diagnosis, it is possible that there may be some maternal contamination that gets into that prenatal sample. So it's important for us in the genetics labs to test for maternal cell contamination of the fetal sample in order to produce a valid diagnostic result. And there are different methods by which we can do this maternal cell contamination testing. Once we have identified the underlying variant in factor VIII or the factor IX gene, we need to classify that variant as to whether it is causative of the haemophilia disorder. So the WFH recommends that variants are classified according to the American College of Medical Genetics and Genomics Guidelines. That's the ACMG or ACGS guidelines for genetic classification of variants. These identify variants as either pathogenic, likely pathogenic, a variant of uncertain significance, likely benign, or benign, according to the evidence that pushes that variant as towards its causative of disease or not. The WFH also recommends that variants are described using the Human Genome Variation Society nomenclature, and this is a standardized nomenclature for describing variants so that a variant that is detected in one area can then be interpreted in a different location. Providing the corresponding legacy nomenclature for the factor VIII or factor IX can also be helpful to the clinician for comparison to previous patient or family clinical reports, as nomenclature has varied over time. After we've classified the variant, we need to produce an interpretive report. And these reports should be clear and concise and contain the following information. We need to provide patient information that identifies that individual as for that sample on the report. So we need to include the patient's name, date of birth, the ordering clinician, the date of specimen collection, their diagnosis by the clinician phenotypically. We also need their baseline factor levels so that would inform our testing. And also if there is a family pedigree available, as that can inform the mode of inheritance of the disorder. Reports should include a description of the assay, and this is quite common to be in the footnote of a report, so it doesn't detract from the message of the report to the clinician. The description should include references to the literature if this is applicable. It should also include the limitations of the test and any limitations in the methodology. There should also be the genomic reference sequence that has been used for analysis, and this should be described on the report, as the genomic reference sequences can vary again over time. The results should be clear and concise and not confusing or conflicting for the clinicians. They need to include the DNA variant that has been described and in which gene, and they need to be described in HGVS nomenclature and using the ACMG variant classification. The interpretation of test results should be in a clear, concise format, and they should be useful to the ordering clinician. These should include recommendations for follow-up testing if that is indicated, and any implications of test results for patients and their family members. And the role of genetic counselling should be stressed that it is important for these patients. It's a vital role that the ordering clinician and the reporting scientist should be able to have communication about those results of the reported genotype and the potential consequences phenotypically for that patient. In the guidelines, there are recommendations about strategies for if a causative variant has not been detected in the genetic analysis. In 0.6% of severe haemophilia A patients, and in 2.9% of patients with mild or moderate haemophilia A, and also in 1.1% of patients with mild or moderate haemophilia B, there is no causative variant detected within the essential regions of the factor VIII or the factor IX gene. In these patients who have a strong diagnosis of haemophilia, but no factor VIII or factor IX variant is detected using current diagnostic technologies, other genetic causes must be considered. For example, deep intronic variants that can't currently be detected. As I mentioned earlier, these might be able to be detected through massive parallel sequencing or whole genome sequencing, but these technologies aren't widely available. For at-risk female relatives of people with haemophilia in whom the familial variant is not detected, for example, in a patient, in the mother of an index case with severe haemophilia A, where the mother doesn't appear to carry the genetic variant, the possibility of mosaicism should be considered. So that's germline or somatic mosaicism. And this should be discussed during genetic counselling. And it's important to highlight this on our interpretive reports as well from the genetics lab. For people with haemophilia A, in whom the mode of inheritance is inconclusive, and in whom no inversion or variant is detected, then other potential diagnoses must be investigated. For example, type 2M von Willebrand disease, where you would have lower factor VIII levels, combined factor V and factor VIII deficiency, or other types of von Willebrand disease. The modes of inheritance of these disorders are different to haemophilia A, so it's important for us to distinguish it genetically. For symptomatic females with low phenotypic coagulation factor VIII or factor IX levels, in whom we only find one pathogenic variant, we might be able to perform investigative tests for X chromosome inactivation patterns. But again, this is only if it's locally available, and this is limited to different areas. Quality assurance is an umbrella term that's used to describe all measures that are taken to ensure the reliability of laboratory testing and reporting. In genetic testing, this covers the whole process from DNA extraction through the analytical process through to the interpretation of the variant and the production of the interpretive report. Genetic diagnostic laboratories should undergo periodic accreditation if this is available by an approved body. Accreditation assesses against an internationally agreed standards, and this ensures a high quality diagnostic service. Internal quality control of genetic tests should also be performed and recorded routinely within the laboratory, and this ensures robustness of these assays and continued processes. Laboratories should participate in external quality assessment schemes for the genetic tests that they provide. And this can be either through a formal EQA scheme or through informal sample exchange between laboratories. I have a few case scenarios now just to reiterate the testing process for genetic testing. The first is the severe haemophilia A case. And the index male has a factor VIII coagulation phenotypic level of less than 1 international unit per deciliter. And there was no previous family history of haemophilia. So according to the flowchart, the first test that we would do is screen for the factor VIII intron 22 inversion mechanism. And by testing for this, we identified hemizygosity for this variant in this patient. And this was confirmed by repeat testing of the DNA sample. And so because we identified the underlying genetic cause, no further genetic testing was required in this individual. So the recommendations on the report were that this individual is hemizygous for the factor VIII intron 22 inversion, and this is consistent with the diagnosis of severe haemophilia A. And this information can be used, if required, in female carrier studies within this family. The second case scenario is a haemophilia B case. And this was an index male with mild haemophilia B. His phenotypic coagulation factor 9 level was 10 international units per deciliter. And so we performed full factor 9 gene screen by PCR and Sanger sequencing. Through this process, we identified a hemizygous in exon 4 of factor 9. This variant has been described here according to the HGVS nomenclature. The NM number is our reference sequence number that was used to analyse this variant. And the cDNA numbering is specified there. So in the cDNA at position 316, a G was substituted to an A. So that's C.316 G to A. This means that at the protein level, the glycine amino acid at position 106 was replaced by a serine amino acid. And that's identified there as P.gly106 serine. We performed classification on this variant according to the ACMG criteria. And as a result, we could identify this variant as a pathogenic variant. This variant was extremely rare in the general population and had been reported multiple times in affected individuals in different geographical populations. There's a low rate of benign missense variation in factor 9, and computational evidence using different software programs supported a deleterious effect on the protein due to this change in amino acid. The patient's phenotype is also highly specific for the gene, so the low factor 9 level is highly specific for the factor 9 gene, so we could apply this criteria as well. And as I say, as a result of all this evidence put together, we could identify this variant as pathogenic. So as a result, the interpretive report, we could identify that this individual is hemizygous for a factor 9 pathogenic variant, and that this is consistent with his mild haemophilia B phenotype. This information can be used, if required, in female carrier studies within this family. For the next case scenario, we had a severe haemophilia A patient who had a factor 8 level of less than 1 international unit per deciliter, who had previously been identified to be positive for the factor 8 intron 22 inversion. We had a sample referred for his mother to determine her carrier status, and she was found not to carry the familial variant. Whilst this reduces the probability that she's a carrier for haemophilia A, the possibility that she's a germline or somatic mosaic for the variant can't be excluded. So the risk of having further affected children is reduced, but can't be entirely excluded. So genetic counselling is extremely advised for this individual. And so prenatal screening is advised for all future pregnancies, and all pregnancies should be identified as at risk. In summary, genotype analysis should be offered to all people with haemophilia and their at-risk female family members, as indicated on the previous slides. Genetic counselling for people with haemophilia and their families is an essential requirement prior to genetic testing to give them all the information that they require and to provide informed consent for the process. Genetic diagnostic laboratories should adhere to strict protocols and procedures to produce sensitive, specific, and robust results. The interpretation of the results of genetic testing should be performed by scientists who have knowledge and expertise in haemophilia genetics. And the ordering clinician and reporting scientist should have a good communication and be available to discuss the potential phenotypic consequences of the reported genotype. I'd like to thank the co-authors of this chapter for all their contribution to the development of this genetic assessment chapter for the WFH for the guidelines. Thank you.